Hi, my name is Nan Sabrina binti Muhammad Shah. I'm from Class C. Today I'm going to present to you photosynthesis from Chapter 2. What is photosynthesis? Photosynthesis is the remarkable process by which plants make their own food. It occurs in the chloroplast of plant cells, where the green pigment chlorophyll captures sunlight. The basic equation is simple. Carbon dioxide and water, with the help of sunlight, are transformed into glucose and oxygen. Plants absorb carbon dioxide through tiny pores called stomata and take in water via the roots using the energy from sunlight. Chlorophyll converts these raw materials into glucose, a sugar, that provides energy and growth for the plant. Oxygen, byproduct of this process, is released back into the atmosphere, playing a crucial role in supporting life on Earth. So, next time you see a plant, remember it's not just growing, it's performing an incredible life-sustaining chemical reaction known as photosynthesis. There are two stages of photosynthesis. The first one is light-dependent reaction, and the second one is Kelvin cycle. The first one, light-dependent reactions located in thylakoid membranes within the chloroplast. ATP and ADPH and O2 are the main product. The second one, dark reaction located in stroma, the fluid-filled space of the chloroplast and produce glucose. The three main factors is Lycott. I use the word Li for light intensity, the second one CO for CO2 concentration, the last one is T for temperature. The first factor is light intensity. I use I and L to remind me that the graph increases and level off. This because as it increases light intensity, the greater photosynthesis increase up to a certain point. Eventually, CO2 or temperature will become limiting factor. That's basically a factor that stops photosynthesis from occurring faster. This is what causes the graph to level off. The second factor is carbon dioxide concentration. I use 2 in CO2 to remind me the graph increases and level off too. But this time, limiting factor that plateaus graph is either light intensity or temperature. Finally, the last factor for photosynthesis is temperature. I use the Re at the end to remind me that the graph reduces at the end. As temperature increases, the particles move around, which result in faster rate of reaction. But this happens up to optimum temperature, where the rate is fastest. At high temperature than optimum enzyme in plant nature causes rate to decrease. There are two types of light reactions. The first one is cyclic pathway and the second one is non-cyclic pathway. The first one is cyclic pathway. The pigment in PS1 absorb light energy, which excite electron to higher energy level. These high-energy electrons are directed to an electron acceptor before traveling through a brief electron transport chain. While moving, their energy is used to pump proton across thylakoid membrane, resulting in a proton gradient. This gradient activates ATP synthase chemiosmosis. After releasing energy, the electron return directly to PS1, then conclude the cycle. The second one is non-cyclic pathway. The light-energized electron in PS2, these electrons then follow an electron transport chain. Water molecules split to replenish the electron lost by PS2, releasing oxygen and proton. As electrons pass through the chain, their energy is used to pump proton, resulting in a gradient that promotes ATP synthesis. The energized electrons reach PS1, are re-energized by more light, and are subsequently employed to convert NADP plus to NADPH. For dark reactions, we have Kelvin cycle. Kelvin cycle occurs in the stroma of a chloroplast and uses NADPH and ATP that we produce in the light reaction. RUBP is a 5-carbon molecule that's gonna bond with carbon dioxide and form the 6-carbon molecule. Rubisco, the enzyme going to set the reaction. The 6th unstable molecule that instantly gonna break down two 3-carbon molecule that is called 3-PGA. The 3-PGA molecules are gonna be changed to something called G3P. This reaction is gonna take place by enzyme using ATP and NADPH. One of the G3P is gonna go on to make glucose. The other G3P are gonna be recycled to make RUBP again. Both of these processes are gonna be fueled by enzyme that is using ADP. This G3P molecule will go on to make glucose, however, there's only 3 carbons here. So, this whole process has to occur twice for one glucose to be made. These numbers in front of the molecule represent how many of those molecules are present. There are three importance for Calvin cycle. The first one is primary importance lies in fixing atmospheric carbon dioxide into organic compound. This is the process where inorganic carbon is converted into organic form like glucose and other sugar. These sugar are the basic building block and energy source for plant and ultimately for almost all other life form on earth through food chain. The second one is Calvin cycle effectively stores energy captured during the light-dependent reactions in the form of ATP and NADPH into the stable chemical bond of glucose, while ATP and NADPH are short-lived energy carrier. Glucose and other carbohydrates provide a long-term, usable energy reserve for the plant's growth, development, and metabolic process. The last one is as the process of organic compound, Calvin cycle form the base of nearly all food web. Plant through the Calvin cycle produce the food that herbivore consume and in turn carnivore eat herbivore. Without Calvin cycle, the flow of energy and matter through ecosystem would cease, and life as we know it would not exist. It also helps regulate CO2 level, which is crucial for maintaining
So
Earth's
that whole
climate.
process must occur twice just to make one glucose molecule. However, G3P is a very versatile molecule that can be made into a lot of different organic molecules for the plant. So our main product of the Calvin cycle is G3P, which later goes on to form glucose. This is an essential molecule for plants and all life in general.
Thank you for watching. That is all from me. Thank you.